back to your college experience. And this is where we're at. Let's just see where that's going. Yep, okay. And so this week we're going to roll on because we're going to finish chapter 17, cell cycle, maybe today, really close. And so you want to read chapter 18, and it's about programmed cell death. And you'll see we'll foreshadow that a little bit more. Then work on your term paper, and that's been described, and that's just happening in the background. People are plugging away at their term paper, picking a receptor of their choice, and asking me questions about it, and just getting it done. Then we have an assignment due today. You can hand it in here, or you can email me pictures of your work to go back over the exam one. Now the whole point of that assignment is to go over back on exam one. And it's not going to change your score or grade, it's just to get you to go back there because it'll help you as we move forward. And then the fourth thing is we have an assignment due on Thursday, and it's pictures emailed to me by class on Thursday of the cell cycle drawing from the cell cycle video. And you've seen before that those videos are just as important as class time, and so I attach an assignment to them just so people are like, okay, I have to deal with that, okay? So that's where we're at on that. And we can just pause here, and it's great to ask questions anytime, but does anybody have any questions right now? Are you sure? Okay, so I think the last time we saw each other at the start, Sam talked about the cellular response, what the T cell does in response to activation of the CAR, the chimeric antigen receptor. And the cellular response included the cell doing a non-genomic response of rearranging its cytoskeleton to hug the cancer cell. And the reason for doing that is twofold. One is to make more contact with the cancer cell and activate more of the receptor. So it's a positive feedback. At first, the cells are somewhat rounded, so only a relatively small number of receptor molecules make contact with the target molecule on the surface of the cancer cell. But as it hugs it, more and more of those get contact. So that's one. And then two, as it's hugging it, it's getting ready to, and we might also just go with a lame analogy, it's getting ready to kiss it, the kiss of death, because it's going to secrete death factors. So first thing is a non-genomic response, changing the cytoskeleton to do that. Then another thing is it's going to activate the pathways that we're learning about, like the RAS MAP kinase pathway, to stimulate the cell cycle. Because if this T cell is activated, there should be more of it. So let's make more of these T cells, including some of the cells that will live longer. That's how our body creates immunity, is to have activated immune system cells divide and then stay alive longer than the typical blood cell. Most blood cells die after a couple of months, just like the way our skin is constantly turning over, and they're dying in part because they haven't been activated, and our body is surveilling the entire environment and checking it out, and if they get activated, it wants to really invest in those cells. So stimulate the cell cycle. Then another thing is to make these death factors. So they're new products. Some of them are proteins, but most of them have to be synthesized by enzymes. So we need the enzymes, which are proteins, which have genes, to be expressed to synthesize and release death factors. There are some of them that just simply make a hole in the cancer cells, so the cancer cell is just going to start leaking to death. Then the last thing that Sam talked about is another thing is that other factors are released from the cell to stimulate nearby other immune system cells to also join the fight. All of those are signaling molecules going from the source cell, the activated CAR T cell, to other target cells, other T cells, other immune system cells that are going to join the fight and help regulate the response. Trying to keep it careful so it doesn't turn into an autoimmune attack on healthy cells, but also really activate it and target it. And those helper cells also secrete kill factors like that. So, it's mobilizing not just that T cell, but other cells to help decide if it's the right thing to do and help in every way. And so we learned about signaling molecules, and I won't write it down because you don't need to learn the term, but the term is cytokines and chemokines. They're just a specialized term for these signaling molecules that go between cells in the immune system. So great. So that'll be an important part of our next exam, is to include those cellular responses. Okay, and there's just an image of those, and if you have a question about that. The next thing I wanted to mention is that that's all a bunch of biomedical research, and that's getting done here in Scarborough at Maine Health Institute for Research. And there are students that do an internship down there each school year. And right now, the applications are getting done for next school year. And I'm the person in charge of that. Sam is doing that internship right now. Joseph did it, and probably other students in the class have done it, and classmates certainly, because we have about a dozen USM students down there every school year. And so you apply in the next 2 weeks for in the next coming school year, and you can learn all about it. There's a video that code goes to explains it. It's a pretty easy application. It'll be 2 hours to put it together. If you're going to be around for the next school year, I'll just mention that this is the best laboratory research training program internship I've ever seen. And before I was here, I was at big biomedical research universities that had a big hospital on campus with many buildings. Like, people tell me I'm lost in the science building. Where I was before this, there were like 15 buildings, like 20 buildings like that, connected to this big hospital. I was lost all the time. Everybody in there was lost all the time. We're just trying to find an ice machine, and we're like, I don't, because they don't bother putting windows in those buildings. They're just like hallways, and you're just like, wow. Yeah, and at UCLA, at Los Angeles, in Seattle, Minneapolis, New York, this is the best, and there's a lot of reasons for it. And so I just keep pushing it, trying to get students down there. It's a really good opportunity. You don't have to have any research experience. You just have to be ready, and you're ready for taking this class. You should tell your classmates about it too, and if you're going to be around, you should apply. The stipend, you get paid almost $9,000 a year, because I just keep asking for more money, because there's some students that can't participate because they have to work their way through college, and I'm like, we have to give them a chance because it's just so good. So if you have any interest in that, let me know, and I can help add stuff to it. Because it's all of it is this stuff. It's all molecular genetics of mice, using transgenic mice to look at all kinds of details of cancer and heart disease and obesity and bone health and all kinds of things in these research labs. The research labs on a daily basis are really similar. They use a lot of similar techniques like CHIP and PCR and things like that. Okay? So we're looking at the cell cycle, chapter 17, and I just included things to kind of go back and remind you after your spring break that, we're done talking about that one, we're done talking about that one, and that's one we always really wanted to talk about, because that's the one that's regulated under a scab stimulating our cell cycle at start, or the UNS transition. So we first talked about that one, the G2 to M checkpoint, and we want to go back to it to remind you, but then we also want to add a little bit to it because you've been working on the cell cycle drawing assignment, and I've gotten that from most students already. And so it's just a simple checkpoint. It's just, if there's DNA damage, we're going to stop and take care of it. And we do that by having inhibitory phosphates on the CDK1. So the cyclin B is there, it's accumulated over G2, it's ready to go. It's not limiting. What's limiting is that we're going to be at this G2 to M, and if there's DNA damage, CDC25 phosphase was not going to remove the phosphates, the inhibitory phosphates. So there's the plan. We got to activate CDK1 to get into M phase. Then CDK1 is the action enzyme all the way through M phase, and it gets turned off at the end of M phase, and at the end of M phase it gets turned off by degradation of cyclin B. CDK1 and the other CDKs are always present in our cells, in every one of our cells, and every cell that's an animal or plant or any other eukaryotic cell on our planet. There are extreme orthologs of all of these proteins in every cell, because if you have mutations in them over the last 100 million years, you just die. Okay, so there's that. We went through that, zoomed in on that because that's the key feature, is to look at those inhibitory phosphates that CDC25 is going to remove. And CDC25 is regulated by this upstream kinase check 1, which is regulated by DNA damage. So that's the circuit. That's the little electrical circuit that controls that checkpoint. You could take a washing machine apart. You could look at the little switch that's connected to the lid, and you'd see the little switch is just a little light switch. And it's off if the lid is up, and it's on if it's down. And it's just a simple block. And this, in case, of course, it's not an electrical story like in a washing machine. It's a biochemical story. And so that's what's happening. Now, the next thing is we were set with that. We want to just dwell on M phase a little bit. I mentioned before that any time in the cell cycle, if CDK1 is active, you go into M phase. The chromosomes condense and become visible. The nucleus breaks down. The chromosomes line up, and the cell tries to divide. If it's the wrong time, the cell will not succeed, but it will try. And so that brings us to some of the talk in the cell cycle assignment, which is to look at M phase a little bit. And these are some old terms that have been around a long time, and are talked about in chapter 17 of the book, and in the video a little bit too. And we just want to make sure we learn a couple of things about each of those. So the exam could just be 2 points, 2 points, 2 points, or 1 point. What's an important thing that happens in prophase, metaphase, anaphase, telophase? That's M phase. And I'll just add another term because it's not part of M phase, but it's part of the cell cycle, and it is called cytokinesis. And it's when the cell divides in two. So that's not M phase. And if you look, cytokinesis actually starts to happen toward the end of anaphase, and then through telophase, the cell is getting everything organized to divide. These are big cellular level tasks that have to get done. And so that's what CDK1 is doing here. Where it gets activated, it starts phosphorylating targets, and then it stops phosphorylating them at the end of M phase. Right? And so we're just going to learn, I think, 2 of the targets, because CDK1 is, and I guess I could write this out, there's a term that we can use, and it's not the world's greatest term, but we can learn it just because it's a satisfying term. You just have to be careful in using it because it's kind of a little bit vague. So regulatory, we've learned about those. We've learned about regulatory proteins like that, right? Transcription factors are regulatory proteins. Their job is to turn things on and off, like that. Yeah. So in these pathways, there might be several regulatory steps. Think about under a scab. PGF, PGF receptor, RAS, RAF, MEK, ERK, ELK, all are regulatory proteins, really. They're regulating that response. Then you get a response, yeah? And at the end of the line, there's a protein, and this is the term, and it's kind of a loose term, but it's called an effector. And those are the things that actually do the action. And so CDKs are, some people would call them effectors. They're not regulating, they're doing the job. And it's a biochemical job, and so we're just going to learn a couple of examples of that because they're very memorable. So here's one thing that happens during M phase. In not all eukaryotes, but lots of eukaryotes, and certainly us, the nucleus breaks down. And it stays broken down during M phase, and at the end of M phase, the nucleus reforms. What's happening to it is that it's still, the parts are still there, they just disassemble, okay? So that's one example of what we want to look at and learn. And the first thing we just need to mention is that to do all of the phosphorylation that needs to get done for M phase, one of the first things that CDK1 does is it phosphorylates 2 helper kinases called Polo and Aurora. And it's just a detail, but it's just such a famous job because these sort of three sisters are going to be phosphorylating all the targets in the cell needed to be in M phase. You know, and why would the book show it? Because it's true, but also because it just helps kind of underscore what a big job this is. You know, the CDK1 is there, it's always there. When it gets activated, it's like, I need help. So it's going to activate these other, Polo and Aurora, and look at their targets. So I mentioned nuclear envelope breakdown. That's what we're going to do, and it's just the nuclear membrane. The nucleus is going to disappear. Again, not in all eukaryotes, but in us, in mammals. So phosphorylation, and we'll write this down, of these proteins that are part of the nucleus, part of the nuclear membrane form, you're going to phosphorylate them, and they just disassemble when they're phosphorylated. So that's the answer to what happens to the nucleus. It disappears because a component of the nucleus structure gets phosphorylated by these kinases. Then here's another one. You can see the chromosomes. They condense. Histones get phosphorylated, so do these other proteins called condensins and cohesions, and we'll go on to slides that have those in there. So that's what that is. So there's that, and then these other ones are not ones that we're going to talk about, but they're more targets, and it's a longer list than that. It's lots of things. The cellular activities really change during M phase in response to these effector proteins, CDK1, Polo, and Aurora. Okay, so here's the nuclear membrane, and on the inside of the nuclear membrane, there are these proteins of the cytoskeleton called lamins. So we'll just write that down because they're kind of like building blocks, and they just make a spherical shape that the nuclear membrane fits around. They're almost like the posts on a tent. You know, when you go camping, you're like, we'll put up these tent poles, and then the tent canvas just goes over it. The nuclear membrane is like the canvas, and then these posts are these nuclear lamins. And so let's just add a word to that. So these effector protein, we can leave that there because we can just say what's their effect. And one is the dissolution of the nucleus. And you know, if your tent is up and you take out the tent poles, the tent is now down if you take out these things. And so we're going to have CDK1 and Polo and Aurora phosphorylate these proteins, and they're called nuclear lamins. And they're called that because the lamins are a big family of paralogs that arose by gene duplication, and they're components of the cytoskeleton, and they are examples of what are called intermediate filaments. So we've talked a little bit about microtubules, and there are microfilaments. These are intermediate filaments. Intermediate filaments are structural, like the 10-stake story, and when these get phosphorylated, they just disassemble. So when they're not phosphorylated, they make this mesh-like spherical shape, and when they're phosphorylated, they just disassemble into monomeric proteins. So they're still there. It's like the 10 sticks are still there. They're just scattered throughout the cytoplasm. Yep. Okay. So that's one example of this effector protein. Is you're not going to have a nucleus, and you could just destroy the nucleus anytime, temporarily destroy it anytime, and in the cell cycle that CDK1 gets activated. Okay. Super duper. So here's another, and that is, it's just showing, and this was back in an earlier chapter, and you don't have to dig it out, but we learned that histones can be acetylated, and that opens up that chromatin. Well, there are lots of covalent modifications for histones. We just learned one. Another one is they can be phosphorylated, and in this case, phosphorylated by CDK1, Aurora, or Polo, and when that happens, they super condense. So when these effector proteins phosphorylate histones, and that's our first choice, the histones compact, so the histones, so phosphorylated histones pack tighter, much tighter than they were in closed or heterochromatin. and the chromosomes become visible. They're so condensed, so compacted. And that's the hallmark feature of mitosis. People have seen that under a microscope for 100 years. They're like, look at all these cells. I can see the chromosomes in those cells. We're going to call that mitosis or M phase. You can see them because they got phosphorylated on their histones. There are additional proteins that get phosphorylated that help in this condensation, but because we know something about histones, that's what make up nucleosomes. We'll start with those. And you can see these effector kinases just going like, you know, and they just, it just forcibly cause the chromosomes to condense. Is there any gene expression during M phase? Nope. You better have gotten that done now, because we are going to condense these chromosomes. We're not going to use the information in them. We are going to have everything ready, and we're going to roll through M phase, because they're just going to be all packed. Now, maybe there's an exceptional gene, but maybe not. Doesn't really seem that there are, but they possibly could be. But in general, the chromosomes are super shut down. Okay? So that's that example. Then, just like it, there are these other proteins, and I think maybe I'll just draw their names on here. You can see them, but they're there in that figure. There are additional protein targets that get phosphorylated, and they're the condensins and cohesions, and we learned about them a little bit because they're looping proteins. And we learned about them, about how we can shorten up the leash, the tether on the enhancer to bring the transcription factor closer to the start site. So that's what these guys are doing. So they've had a role to play. And when they're phosphorylated, they just play that role much more dramatically, and they just loop up all the DNA, and again, help the chromosomes condense. So histones, cohesions, cadensins, all parts of chromatin being the DNA plus the proteins that are associated with it. That's what chromosomes are made of. We see the chromosomes during M phase because they have gotten target proteins phosphorylated by CDK1, Polo, or Aurora, and they just become visible. And they stay visible until the end of M phase, when CDK1 gets broken down and a phosphatase starts removing those target phosphates. We don't need to learn about that phosphatase. Okay? So, just like that. Okay? And there's more to this chapter in the book. The book talks some about some cell biology, talks about the microtubules in the spindle. There'll probably be a couple of questions on the test or the final about a little bit more of the cell biology of it. If you have questions about them, bring them to class. But these are the major highlights in M phase. We have these effector protein kinases doing dramatic things in the cell, and just driving it through M phase. Great. Okay. In the video, I also distinguish between mitosis and meiosis. They're just two different real ways of having to deal with the cell cycle. Had a couple of questions students have emailed me about meiosis, just to clarify that. Great. As I talked about in the video, I won't add to it here. So that was, back to this, regulation of entry into M phase, and it's all about CDK1, and then that was just about, well, what does CDK1 do? And it's just super satisfying at the level of car mechanics, where you're like, well, do the tires do anything else? The car mechanic is no. They just help the car roll. That's it. You know, done talking about tires for our car mechanics course, like that, okay? So we've talked some about the regulation of start, and we'll kind of loop back over that. How do we organize the regulation of start? Well, it's pretty complicated, and so I like this visual. There are really three big players. Again, the CDKs are different. They're not CDK1. We don't need to learn the numbers. It's just other CDK1s that are the most prominent at start. And they get regulated by these three major ways. There are cyclin-dependent kinases, and sometimes there's not enough cyclin. So the light is red. The CDK is inactive because it's rate limited by not enough cyclin. Did we learn about that? Yeah, that's the story in a scab. Under a scab, the skin cells are rate limited in their cell cycle progression as they're at start, the G1 to S transition, the restriction point, and they're waiting for light to turn green, and they're waiting, the light is green because there's not enough cyclin. we're going to physically increase the expression of the cyclin genes using transcription factors like MYC, and we had a little mention of June and Foss, and we'll repeat that again today. So we have some options to activate cyclins. And then there are these new things, these small inhibitory proteins, and that's another one. And the third one is phosphorylation, both activating and inhibiting it. We're not going to talk about any examples of the phosphorylation stories because we have such a good one over on the other side of the cell cycle. There's just more of it over here. It's different and it's complicated. Great, you could figure it out, but it's plenty for us to just talk about these other two features that are really often the rate limiting step in the G1 to S transition. Okay? So there they are, examples of CDK and regulation at start. Cyclins, phosphorylation, and binding of inhibitory proteins. There's a bunch of inhibitory proteins. We talked about that before break. We're just going to learn about two of them, P21 and P27. So in that context, we're going to have 5 examples. The first one we started talking about a little bit. That example is about just us learning about what if the cyclins are rate limiting. Well, we're going to have to activate expression of more cyclins, so it's going to be a nice, satisfying transcription factor gene expression story, like we learned about MYC. So we're going to add to that. We already did some on Thursday before break. Then this one is just introduces a huge new concept that the book dives into more than we'll be able to dive into in class. Then you can see there are 2 of these small inhibitory proteins, and then there's another protein here, and we'll see how these examples tie together. And they're just selected. They're all covered in the chapter, and it's more, but these are like the highlights to help build a framework for you to learn and hopefully remember these. So the first one is genomic response. So we're going to activate new gene expression. We're going to increase the synthesis of the cyclin genes. And that's what MYC does. So in this pathway, we learned last time that phosphorylated ELK, and now we also learned about serum response factor, both active and stimulate the transcription of lots of genes. But for us, especially memorable is targets of cyclin genes. And if we make more cyclin protein, the light will turn green. The CDKs are cyclin-dependent kinases. They don't have enough cyclin to go into S phase. Yeah? So there's the story from the discovery of serum response factor that we talked about like we go last Thursday, and they call that the restriction point. So now we have different terms. We have the G1 to S transition, we have start, we have restriction. They're all the same synonymous, fully synonymous terms. Great. This is just showing some targets of MYC to emphasize there are a bunch. People learned about the targets of MYC using that CHIP strategy, the chromatin immunoprecipitation. They pulled down all the MYC target genes, and it includes cyclin genes. You can see from this, though, that, START is pretty complicated, right? So we're just distilling it down, and we're not oversimplifying anything. We're highlighting the big features of it. You know, like it's not oversimplifying a person to say, well, a person has a head, arms and legs, body. You know, that's not oversimplifying, just beginning to say now, we're going to focus on the head and talk a little bit about the head, you know? And so the head being like activation of CDKs. And we know that MYC does that. And then here's the book, and the book actually chooses to focus on a different famous transcription factor, and it's called AP1, and it's made up of a heterodimer of 2 proteins, June and Foss. And it's just the same kind of story as MYC. So this is a nice image from the book. It looks just like the MYC story. And June and Foss are famous oncogenes. Like MYC, they're dangerous, and they're regulated a whole bunch of different ways to try not to make them when they're not needed. Yep. And we just didn't pick on them. We picked on MYC, but we could pick on June and Foss. So here, let's just draw a little drawing of this, and we'll build on this. And that is, so there are several cyclin genes. And as just a memorable visual, we're just going to draw one sort of generic cyclin gene, and we'll just call it cyclin. And there are several, so these probably wouldn't be true for any specific one of them. but we're going to say it's true just to help, in the learning. And we start out and we say, yep, that's the cyclin gene that we learned about when we learned about MYC. So there's MYC, and by itself, MYC could stimulate transcription of cyclin. And that would make cyclin protein. I guess we should draw that in too, just so we see that. When it gets activated, we're going to make the cyclin mRNA, and then a ribosome is going to grab that, and it's going to translate that into cyclin protein. So that's how we're going to get the cyclin made. And we get enough cyclin made, and we go through the start. The light turns green, we go through the start G1S restriction point transition. With that, okay? So what we just added here is we said, well, actually, there's another enhancer on this gene, and it is an enhancer that binds this heterodimer of June and phos. And that's fine. And that's what these, that's what regulatory regions of genes are like. They can have multiple activators or repressors of transcription. When we started our course, we just talked about myogenin MYC. That was a really good starting point. Just talking about one transcription factor binding to the enhancer upstream of myogenin and activating it. But almost for sure, there are multiple. And we don't have to learn about those because there are more important things to put into your, brains, and of course, than just a long list of here's more and more and more. Okay. So let me just show you one more because our book talks about it, and that is that another one is that we'll see from a figure in your book, and this is the last one I think we'll learn about is, here's another enhancer, and this one binds beta-catenin, which we learned about for our first exam, and we have to learn more about it for our second exam. Beta-catenin can also stimulate the cell cycle. Beta-catenin is a world-famous protein because orthologs of beta-catenin are found in every animal on our planet. And it does lots of different things. It does lots of different things during embryonic development. And just about every different organ we have, beta-catenin played a big role in the early embryonic development. And it can include stimulating the cell cycle. So that's part of embryogenesis, and it can do that. So that's our drawing. And maybe we'll add another one, but that's, oh no. Yeah, USM is shut down again today. What do you think happened there? I guess it's open again. Still recording, all right? So let's look at that. So you don't need to jot this down, but we learned about this for our first exam. There's Brazil, beta-catenin gets stabilized and overwhelming the TCF inhibition. And look, in this chapter, they say targets can include MYC and cyclin, D1, whatever, like that, cyclin, right? So MYC can do that, beta-catenin can do that. So lots of different ways to activate cyclin. In different cells at different times can stimulate normal proliferation. Our book likes to put little exploding things around proteins that can be causing cancer. And anything that stimulates the cell cycle could potentially get mutated and contribute to cancer. So beta-catenin, June, phos, MYC are all called oncogenes. Genes that, when mutated, could contribute to the development of cancer. Study of cancer is oncology. Yep, all right. So on we go. And so I wrote this to remind me to draw that cis-regulatory region of a cyclin gene at G1 to S. And you could draw two pictures of it, one where it's inactive, one where it's active. Let's just go back to that and see that I kind of did a shortcut there on thinkingly and show you that what I forgot to include is that beta-catenin actually binds on the back of TCF. And TCF, remember, is an inhibitor. So, and so sometimes, often, the interplay between transcription factors on the regulatory region of cyclins can be complicated. I don't know if TCF could inhibit activation by MYC. I just don't know. But it would be a competition, and other scientists know, I'm sure. And But TCF is there. Other ones are there too, activating and suppressing. And gene expression and transcription are kind of like majority rules type of thing, where there are some transcription factors trying to activate and some trying to repress. Yep. Yeah, but that again is the beta-catenin, and so we have kind of a complicated drawing that we could draw about how one gene can have a complex regulatory region. And probably most genes have complex regulatory regions, and the cyclin gene's regulatory region is more complicated than we've drawn. But that's a lot, and it's good. So that's where we talk about transcription and activation of our favorite example, cyclins. But we saw MYC activates other things that are important for going through the G1 to S transition. So that's that example, and we're adding to what we learned from the first exam. And so that's one reason we went back over the first exam, is because you don't have to study all of the old material from the first exam, but if you have any, confusion about the MYC, and you want to go back and make sure you understand the role of MYC. And if you have any confusion about beta-catenin, go back and look at that again, because we're building on that. So again, you don't have to study everything from the first exam, but stuff that we're building on, go back. And there we're building on transcription regulation. And we're really good at being able to draw a gene. Our big focus here is the cyclins. Draw that gene and show how it gets regulated at the level of transcription, genomic response. Okay, so here's this protein, retinoblastoma is how it's pronounced. I should mention that you're perfectly welcome to call it by its perfectly appropriate abbreviation, RB. Retinoblastoma is a gene that codes for a protein, and we'll see it's a transcription factor. So it's all new to us, but we'll see it plays a profoundly different role than the oncogenes we've learned about so far. Retinoblastoma is important in certain kinds of cancers. That's true for all these. And we've seen little hints of that, right? Estrogen receptors are really important in certain types of breast cancer. Retinoblastoma sounds like a problem of the eye, and it is. There's a cancer in the eye called retinoblastoma. And the retinoblastoma gene is mutated. So I'll give you an example of that, and it's from when my family and I lived in Seattle, so it's nobody that you know. And our two daughters, they were really little then, born in toddlers in Seattle, and we knew a family that also had two girls pretty much the same age as ours. And we hung out with all these different families. That family had these two girls, and the younger girl, I'm the kind of person that I don't talk to people about this and ask them, like, what's up with her eye that was just closed, always closed. Just like gently closed, like she was sleeping. All the time. And, super cute little kid. She was, probably 3, 4, 5 as we knew her, hanging out with her. And her family were very, very nice, and they were always kind of in shock. And I think it was because of that. At one point, the dad mentioned to me off to the side, says, she's an RB carrier. And I don't know for sure, but what I think that meant is she was born with 2 RB genes, one from mom, one from dad, and she was born with one, we don't know which parent, that was mutant, that wasn't working. So in her eyes, she had danger. Because if she picked up a second mutation in the healthy retinoblastoma gene, she would be vulnerable to this eye cancer. And I suspect what happened is that doctors knew that when she was born, and they were just monitoring her eyes carefully with eye exam, and they removed one of her eyes. And what about the parents? We don't know which parent, but I think the parents, one of them, was also that, and they just hadn't had a mutation, because mutations are random, right? And they had lived all the way to adulthood without having that mutation. But if their other mutant, their one good gene got mutated, they would get good chance for retinoblastoma too. So just a very specific example of a very difficult situation, right? All the time, every day. There's, worried about that. So here's how it works. You see what it is? It's a transcription factor that's a co-regulator. And we know about that. It's either a co-activator or a co-repressor, and it does not have a DNA binding domain of its own. It binds on the back of another transcription factor called E2F. And it's a co-repressor. So E2F is trying to activate transcription. It's an activator of transcription, and it has hundreds of target genes. And retinoblastoma is on it right now in our cells, repressing it, saying nope, nope, nope. And we see what happens is that if retinoblastoma gets phosphorylated, it gets turned off. Who would phosphorylate it? The answer is that normally CDKs. This is like the off position, and when you turn the light green, one of the most important things is, in this case, is the CDKs at start, they're effector proteins also, and they will phosphorylate retinoblastoma and other targets, and now E2F will activate. So two things we want to be aware of is this is suppressing the cell cycle when it's good. And most mutations, random mutations, most of them damage the gene, decrease the protein activity. And so this is dangerous, right? Because if we damage this, it won't work, and E2F will just be like, let's go. And so the first thing is we have a new term, and that is retinoblastoma is called a tumor suppressor. So we have these two different categories of genes. Oncogenes are genes that normally stimulate cell growth and the cell cycle, and tumor suppressors are genes that normally inhibit the cell cycle. And retinoblastoma was first of those found, and it's famous and it's very accessible. You see what it's doing, that's what it's doing. So when the light is red, retinoblastoma is active, it's inhibiting E2F. And to turn the light green, we're going to activate CDKs, and they'll phosphorylate that retinoblastoma, and it will let go, and E2F will do its job. So that's the first thing. That's all that retinoblastoma does. It's just a little molecular switch regulating E2F. And it probably has a few other targets, but this is the super famous one. So then we turn our attention to E2F, and we say, well, what's E2F do when retinoblastoma lets go of it? And I said it has hundreds of targets. E2F is actually a family of genes. The G1 to S transition gets complicated really fast, but we'll just focus on the idea that it's E2F. It's E2F. So let's just look at that. E2F transcription factors are downstream effectors of the retinoblastoma protein pathway and required for timely regulation of numerous genes. What do? So this is what we want to know. They stimulate the cell cycle. So this is another example of the biochemistry of what it means to turn the light green. The light is green. What does that mean? It means you're going to start trying to make lots of DNA. That's S phase. What do you have to do that? Well, you need a lot of DNA polymerase. So almost for sure, the E2F is sitting upstream of the DNA polymerase gene. And we need a little bit of DNA polymerase during the cell cycle to kind of repair genes and stuff like that. But when it's S phase, we need tons. Because there's also basically no gene expression during S phase. The cell turns its attention to S phase and says, we need to replicate the DNA, there's a lot of it, and we're focusing on that biochemical effort for now. Takes a couple of hours, a few hours typically, to go through S phase, because it's just a big job. And we need a lot of the guys that are going to get done. So E2F is going to activate things like DNA polymerase. Then it's going to activate enzymes that synthesize the AGCT nucleotides that DNA polymerase needs to get the job done. You need huge amounts of those. They're not just hanging around. So it's switching on all the DNA synthesis machinery. since we're going to make mistakes, so it's going to crank up all the DNA repair mechanism. And there are other proteins that are playing a role in this, but this is a massive genomic response to going through the G1 to S transition. And I said before, you know, people will draw the G1 to S as if it's a little bit before start. And this is the reason why, is it's like we're just going to crank up. But we're going into S. When we get through, when the light turns green, we'll be going into S. It's just a question of maybe an hour before we can just crank up all the machinery we need to really roll. So, essential for DNA replication and cell cycle progression. Several laboratories have used genome-wide approaches to discover novel targets of genes like E2F. What kind of approaches do they use? CHIP, chromatin immunoprecipitation, to pull down all the targets of E2F, leading to the identification of several hundred such genes. They're involved not only in DNA replication and cell cycle progression, but also in DNA damage repair, apoptosis, differentiation, and development. These new findings greatly enrich blah, blah, blah. So, you know, we're focused on, well, what does E2F do? Well, it gets the, you know, S phase going. But S phase is all about normal development and growing and differentiating and you know, making tissues and stuff like that. So the E2F genes, proteins play a broad role in that, okay? Here's, you know, just a list, right? From CHIP, these people are, oh my gosh, E2F binds to a lot. And remember, it's a family of proteins, lots of targets. And retinoblastoma plays a big role in repressing that. We don't need any of that junk, you losers, you know? We're not going camping today, so start putting the camping stuff away, you E2F kids. Like that, right? Keeping up. Role of the cell cycle. E2F family members play a role, major role in the G1 to S transition in mammalian and plant cell cycle. So very conserved story, like that. Analysis review unique set of target gene promoters, and they mean that broadly, the cis-regulatory region among E2F members, suggesting that each protein has a unique role in the cell cycle. Among E2F transcriptional targets are cyclins, CDKs, checkpoints, regulators, replication points. Nonetheless, there's a great deal of redundancy among the family members, blah, blah, blah. You know, just a big effort, right, to get out all the camping gear to go into the S phase camping trip. Yeah? E2F. E2F is a group of genes that encodes a family of transcription factors in higher eukaryotes. 3 of them are activators and 6 others are suppressors. So that story gets complicated if you start diving into it. It's a gene family and we're focused on the activators, but others could be repressors. So complicated stuff. All of them are involved in cell cycle regulation, synthesis of DNA. So we don't need to dive into that any further. Nice picture, I think I have it again here. Nice picture. Building off what we've learned in our course, you're like, okay, famous tumor suppressor, famous transcription activator at the G1 to S phase transition, and it's just like, that's satisfying. Because we can keep talking, oh, the light turns green, but now you have a hint. It's just a biochemical story. And it's not, there's no like, oh, and then there's magic. It's just click. We got to go into S phase. What do we need? Yep.
Can we dive into that a little bit, Dave?
Yeah, as we party, because we're still breaking on springtime.
So we talk a lot about how there's like a biochemical story here, right? And so we can expect like there's going to be like a percentage of RB that's phosphorylated or unphosphorylated, right? Like,
Okay.
And that means that we're going to get some expression of this like E2F, but it seems like we're talking about cell processes that aren't like do a little bit, do a little bit more. It's like go, don't go.
Right.
And I'm wondering, how do we make that jump from like processes that are on a continuum to stepwise?
Thank you. And I will answer that question, and I'll show you that by saying, I'll just jump ahead to this, and we'll just look at this, because this is just one example to address your question. So we have a little bit more to talk about that, but we'll just go to this third one. It's very brief. There's just one figure in the book. It's a really simple story, but it underscores what Sam's asked about. So one of these inhibitory proteins, P27. So remember, and I don't think I have it right here, but remember that that's an example, P27. So it can just be like, nope. The cell could be like, I don't want to go. Sometimes I say, P27 is like a school patrol person. So there could be a red light, green light, but even if the light is green, if there's a person who comes out in the street with a flag, school patrol people, if you know what that is, cars have to stop. It just blocks everything. So P27 is like that. It's like, nope, 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 nope. These little kids have to cross the street. I don't care if it's green. You have to wait. See, so all the other stories don't matter. You're like, I'm late for work. And that school patrol is out there being really careful, okay? So it's a very simple example, and look what it shows. And it's a little bit confusing in the book, but we're talking about this context of how do we really switch it? Because all it shows is this. It shows, here's P27, and it's inhibiting the cell cycle. And then it shows us, and for students it's confusing, it shows that if you get some of the CDKs activated, so the light starts to flicker green, one thing that happens is they'll phosphorylate P27, and P27 will get destroyed. So all that is it's a positive feedback loop that you're asking about, and there's lots of that. There's lots of negative feedback loops and lots of positive feedback loops to turn it into a really crisp, grand switch. And all this example, and I made it as a separate example to show you, is, when the red light turns green, initially it's flickering, and you're in your car with your friends, and your friend's like, is it green? Is that green now? And you're like, I don't think so. I can't tell, because it's just flickering. But then it switches. So there's tons of that positive feedback loop to say when it's going to flicker red or flicker green, we're going to turn it bright green. And so there are many, many of the examples. And this is a good one just to explain that confusing figure. What it showed there is just an isolated example of how the light switches to be fully green. And we're done talking about that, right? So you can see it's one of these inhibitory proteins. It's there, the light is red, like that. The school patrol light is just, nope, we're not going. And it starts to flicker, starts to think maybe, and it just switches to be fully green. And there are many, many examples of that. And those kind of feedback loops get confusing really fast, right? Because you just learned about them. You're just like, I just learned that was an activator, but how's it working now? And isn't it weird that it's an inhibitor, but that the CDKs actually can destroy it? That seems kind of like a paradox, like that. But so to answer your question, and there's examples of that, but that's just a very good one for us to kind of just focus on. Now let's just go back to retinoblastoma and show you this kind of confusing figure, like that. And it has to do with the idea of that we're going to turn the light green, and we're going to go. And when the DNA starts to get copied by DNA polymerase, there are several points along the chromosome where the DNA polymerases start. And I'll just write this down here. So the DNA is long, millions of base pairs long, and we got to get this job done. So we don't have DNA polymerase start at the end. There are a whole bunch of sites randomly located along the chromosomes, and they're called origins of replication. And that's a great word, replication, because they're talking about S phase, DNA synthesis, and they're just like, we're going to get this job done. We're going to have DNA polymerase molecules binding each of those and get going. Yep. And so the book is going to show that. And so we just talked about how E2F has lots of target genes going to be activated. And I said, make more DNA polymerase. Make the enzymes that make the nucleotides, AGCT. So another example is to get these origins of replication going. And so I'm going to zoom in them a little bit because there are kind of two parts of that story. And so we'll just look at this. And it's hard to see, but the book does a good job describing it. And it's a pretty simple thing where we should note that we're getting these proteins called MCM. And we can talk about them, but they need to show up at the origin of replication. And so more of those are getting made. And our figure doesn't show this, but it's true that there aren't very many of those around, and we're just gonna make a lot. So another target for E2F would be these MCM genes, to make more MCM protein, because it's gonna be at the origin of replication, and it's gonna help the DNA polymerase find that and get started. Here's the thing the book is emphasizing is, yeah, not only do we need the MCM proteins made, but we also need to have them phosphorylated. So it's another example of the kind of complexity of making this thing green, is we need more and we need to phosphorylate it. And who phosphorylates that are the CDKs. So the CDKs at start are also effector proteins. They are causing the cell to go into S phase by making more of the components, stimulating more of the components to be produced by E2F, but also by actually phosphorylating some of the components to fully activate them and say, go, go, go. So that's the story here, and the proteins are just called MCM, so it's another protein we would learn about, and if you wanted to, you could draw a little drawing like this, and you could say, well, where's an MCM gene, right, like that, and where we are in our course, we would say, if this is an MCM gene, I suspect that there is a E2F protein here repressing it. No, he's trying to activate it, sorry, trying to activate it, and there's probably a retinoblastoma protein here repressing it. And when retinoblastoma gets phosphorylated, it will leave, then E2F can activate it, we get a bunch made, so we start making a bunch of MCMs, but then they get further stimulated by having the CDKs phosphorylate them. So we're gonna make them and activate them, combination. Another kind of example of sort of like checks and balances, where we're gonna make more protein and then we're gonna phosphorylate it to fully activate it. So just a figure like that, where the book shows that biochemistry, again, like Sam talked about, and the switching that's happening here. Super, okay? And so that's retinoblastoma story. Tumor suppressor, and it's inhibiting E2F action. E2F is trying to activate DNA polymerase gene, enzymes to synthesize components of the nucleotides, DNA repair genes, MCM, and lots of other things. Switching on lots of stuff. And so MCM is a good example of something where you're like, I see why that's needed. Right? Got to find all those origins of replication and get DNA polymerase there. Okay? So then we already talked about P27. So let's just talk about this other story, and it's actually, these two proteins will go hand in hand. So P21 is another inhibitory protein, and what P53 is it's a transcription factor that can turn on P21. So now, with the analogy, and we could have that thing where students get up and portray this. We should, but that was so much fun, and this would be fun too. Because we'd have some students playing children, and then we'd have somebody being like an old man, school patrol, and then P53 is like a parent who's waving goodbye to their kids and sees the old man kind of sleeping and says, wake up and get out there and stop the cars. So it's a little bit too much. But it turns out P53 role is to stop. And it does that by getting the P21 made. So it's going to be a really simple drawing that looks like this, where you're like, okay, I see why we learned about genes in this course. So it's a really simple drawing where this is the P21 gene, and if you make it, the protein will be the stop sign. It will be a small inhibitory protein. And who can make it is P53. And P53 gets activated by DNA damage. So we mentioned that start is a little bit challenging because it is a control point. We're just trying to decide if we should go into S phase. but it has built into it also a checkpoint, because you should not start copying DNA if there's DNA damage. So that's what's going on here, is that just a simple biochemical pathway. Try not to confuse it with what we learned about over at G2 to M, because it was a different story, but it was also regulated by DNA damage. So it's a new story, DNA damage, and that's P53's role right there, okay? So a nice visual. And P53 was the second tumor suppressor gene. So it's got a function to stop the cell cycle. So its normal job is to suppress the cell cycle, right? Because it's going to be like, there's DNA damage, we're going to stop. And if you mutate it, you're not going to pause for DNA damage, it's just going to roll on. The tumor will just get more and more mutations faster and faster if we destroy so inactivated in many cancers, it's not, like the early cause of cancer, but as the cancer progresses and gets more and more worrisome, there's a really good chance that there'll be a mutation of P53, because then there's just more and more mutations that are in the cell, because the cell is not stopping to repair DNA damage. It's just like, sure, why not? And so that's what it is. So P53, encoded by the PTP53 gene in humans, is commonly referred to as the guardian of the genome. Ta-da! And I have a little, I think, there it is, guardian of the genome. Like Guardians of the Galaxy, that's what it's based on. That movie's based on this protein. Yep. This is one of the 10 most studied proteins on our planet. People are they're just enthralled by P53. Yep. Due to its activities directed at maintaining genomic stability through repair of damaged DNA and integration of cell birth death signaling pathways DNA damage checkpoints. Right? It's a transcription factor that binds to specific P53 responsive elements in the perimeter of target genes. Its interaction with the genome is complex. So again, it's a complex story, but we have a really great example here. It's going to bind to an enhancer upstream of P21, and it's going to be like, make it. And so here's the image from our book. And it's shown here where normally PP3 gets getting destroyed in the cells, but if there's DNA damage, it gets phosphorylated, that stabilizes it, and it binds and stimulates P21, which is a small inhibitory protein. That's a more complicated figure than it needs to be, but it's still a pretty simple figure and a nice molecular switch. So that's that. I'm glad we got this far, just because then we can talk about the guardian of the genome in a concept chat. So have a concept chat and be like, we can talk about spring break, but I think P53 is pretty cool. I want to talk about that a little bit. Go. Because we have to do this. And if you have your assignment, you can either email it to me or hand it up here, and these are the corrections for exam 2. Look at, I have this slide, you guys. If you don't know what a school patrol is, there you go. Raise your hand if you were school patrol in school. I was. I got in fights with it. I used to just bad. Hit kids with it.
We talked about the regulations
We're just doing another minute. And then we'll check with Deborah. Deborah, what's one thing that your group talked about? Yep, the eye cancer story. So that's a retinoblastoma story. Retinoblastoma protein plays a big role in lots of different cells. It just must be that the checks and balances in the eye cells are extra kind of sensitive or dependent on the role of retinoblastoma. And so, that can be confusing for students to understand, is that it can have a broad role, but maybe in some other cells, like maybe the stomach, there's just more checks and balances that have to be in place. There's less dependence on the role of retinoblastoma. So if it gets mutated in the stomach, maybe less likely to develop into cancer, like that. And so, when we see that, and we kind of got a hint of that with the one with skin cancer, right? Where it's like, ooh, shoot. Those cancer cells are normally very responsive to PDGF, right? That's where it plays a big role. Does PDGF stimulate the cell cycle everywhere? No, but it's all set. They have the cell, every skin cell in our body has the PDGF receptor all the time, because what if you get cut nearby there? You gotta, heal like that. So it's like that. So the eye must have a particular sensitivity to this. And so it was first discovered there. They're like, what is causing trouble in these retinoblastoma tumors? And they're like, well, we found this gene, and we're going to call it retinoblastoma. And they found it was a tumor suppressor style gene. Yep, great. Okay, and Mallory, what's one thing your group talked about?
We just talked about P53 and how it functions with P21 as regulation.
Yep.
Almost, and because I've heard of P53 before, but I didn't have a super solid understanding. But this helped me understand how it.
Yeah, and that often happens, right, and that's part of learning, right? Because there's some proteins that are like in the newspaper, in the news, or you'll hear of a family member, they're like, oh, I have a mutation in people, like that kind of stuff. And you're like, oh, I kind of think I understand it, but yeah. And it's usually a thing where you're like, probably pretty complicated. It gets complicated, but there's a framework where you're like, okay, it's a transcription vector. Does it do other things? No. It's just a co-repressor. It probably represses some other transcription factors in addition to E2F, but it's definitely a big time E2F repressor. And E2F is just a super rowdy transcription factor with so many targets, And it's a great example of why some proteins have lots of targets, is because they have lots of things they need to switch on, right? Like that. So that's a good example. Yep. Yeah. And Juliet, what's another thing your group talked about?
We really went heavy on the P53.
Yeah.
I guess how the start of it is through DNA damage kind of activates it.
Yep. Yeah.
That's different than the entry into mitosis G210.
Right. Yep. Because in this situation, normally if there's not DNA damage, the P53, this very important protein, is just getting destroyed. We don't need you around. Go back inside. Like that. But when DNA damage appears at that point, then it activates that. Yep. So do people have questions about anything at all? Yes.
Kind of to follow what we were just talking about. So it was said that P53 is inactive inactivated in cancers? No, is inactivation caused through mutation generally?
Oh, when it's plays a role in cancer?
Yeah, so it says like it's inactivated in cancers.
Yeah,
Is
that's a
that-
good-
That inactivation or like a mutation creates that inactivation.
That's a good question, and the answer is there's kind of two levels. And so one is the idea that we're going to inactivate p53's function. Well, we could do that through a mutation of p53, like that, right? And it would not be functioning, you know? And that's common because, again, random mutations are likely to inactivate something, you know, like that. So that's what most people would say. They don't just say, they say, like you said, inactivation of p53, because you could have upstream, mutations in upstream genes. Like, for example, what about a mutation in the gene that codes the kinase that's normally phosphorylating p53 and targeting it for destruction, right? Because that's its normal role. Normal situation is it's getting inactivated, it's getting destroyed. So in this case, the story of p53 might be an inactivation either through direct damage to the gene and the protein itself, or indirect by damage to the upstream destruction pathway that normally is destroying p53. Am I saying that right? Maybe not. I don't remember. I was on spring break too. But yeah, but the idea that it could be, the answer is, yeah, the inactivation could be a direct story like that, or an alteration in another gene that's playing a role. Yes, yeah, but in both cases, it's the idea is its normal function is to suppress the cell cycle, so it would be a tumor suppressor. Yep. Other questions people have? We have like another minute or two.
I just have one. In
Yep.
chapter 17, they talk a lot about like yeast and like yeast cell cycles. Like, what do you want us to take away from that for this course?
Yeah, so the key thing is that yeast are a model organism, and why are yeast a model organism? And the answer is because they have orthologs. Somebody would say, oh, they're really easy to grow, and it's really easy to study the cell cycle, because that's kind of almost the only thing yeast does. You know, when you take dried yeast and you add water to it, you add it and it makes the bread rise, you're activating it, you're getting to start to grow and stuff and start to, you know, metabolize. And it's pretty simple, you know, simple little story, single-celled eukaryote, you know, like that. So it's a great organism to study genetically like that. And but the key thing is, yeah, it turns out it was a good choice to study, because even though it's super distantly related from us, the genes that are mutated when you're studying the cell cycle in yeast are genes that are orthologs in us and every other eukaryote. So that's the key thing on that. The reason it's in the book is because the people that did that work are still alive, and they're good friends of the author, and it's significant, you know, because not just out of a friendly way, but it's significant to dive into that story, because it's still pretty modern research. You know, the tools they used are pretty modern tools like that, and so it helps kind of set the context. But there really isn't specific stuff that we need to learn about yeast. Yeah, yeah. You saw mentioned like p53, E2F. Some of those stories are not actually orthologs. The big regulatory stories are like the CDKs and the cyclins. Those are orthologs in yeast. But there are some regulators of the cell cycle, especially at start, that are found not in every single eukaryote, because there have been evolution of regulation of the cell cycle, you know, as organisms got bigger and more complicated. Somebody asked me in the lab class, and this will be the last point I make today, somebody asked in the lab class, maybe Justin, were we talking about whales?
Yeah.
Yeah, so scientists are like, hey, whales are great big mammals, and they live a really long time, and they don't, when you know, catch a whale, you don't see much evidence of cancer in it. So people have turned to other organisms with that idea of, well, whales are not going to become a model organism like yeast. Goodness, we can't graze them in a lab, but we could actually get a little tissue, and we could ask things like, well, how many copies of the p53 gene do they have? And scientists have found that, I think it's p53 specifically, has gone through a bunch of gene duplication in big whales to be more protective, right? So again, Mariella asked about inactivation. Well, you could mutate one p53 gene, but what if you have a whole bunch of p53 genes, right? You know, it's pretty well protected from that kind of, you know, damage like that. So, you know, it can be fascinating also to go out into what people would say is a non-model organism. You know, it's not a fruit fly, it's not yeast, it's big whales, and say, well, that's the diversity of life on Earth. How do those guys deal with that? And you can go intentionally looking for the p53 gene and ask about, do they have it? And people are like, yeah, they do, and they have a whole bunch of copies of it, you know, like that. So that's where we're going to stop today. In fact, we'll finish in the first 5 minutes on Thursday, chapter 17, and go on to program cell death, chapter 18. Yay, welcome back.